Hi, my name is Wael Abdrabou. I'm a PhD student at New York University. During the blood stage of Plasmodium falciparum malaria, the serum becomes home to byproducts of parasitic metabolic reactions and host immune response. Understanding the changes that takes place in the metabolome in vivo could provide us with a better understanding of the reasons behind inter-individual and inter-ethnic variation in host immune response to infection. In this study, we used high-resolution metabolomic and transcriptomic profiling of 153 children from two different ethnic groups in West Africa, the Guan and the Fulani. One of these two ethnic groups is known to be less susceptible to malaria. However, the mechanisms behind this remain poorly understood. Through high-resolution metabolomic profiling of the Guan children, we were able to identify 667 metabolites. And by contrasting the metabolomes of these children before and during infection, we were able to see the overall effect of infection on the serum metabolomes. We were also able to identify differentially abundant metabolites. Among these metabolites, we identified a set of endogenous steroids. The abundance of these steroids is significantly associated with higher parasitic load in these children. Another intriguing part is that the concentration of these steroids and the enrichment of these steroids in the serum showed to be also negatively correlated with the lymphocyte count. To investigate the potential immunosuppressive role of these steroids, I used integrative metabolomic transcriptomic analysis. The analysis showed that the steroid abundance is significantly associated with the expression of genes implicated in T-cell signaling pathways. Pathway enrichment analysis using these genes showed that the enrichment of these steroids is associated with an overall inhibition of T-cell function during infection only. When we looked at the steroids response to infection in the Fulani group, who are less susceptible to malaria compared to the Guan children, we realized that the Fulani are showing opposing steroid response to infection, which could mean that any potential immunosuppression effect associated with these steroids on the adaptive immune response to infection is not likely to take place in the Fulani children, which could contribute to their lower susceptibility to malaria. To investigate the immunosuppressive role of these endogenous steroids, we isolated PBMCs from 10 healthy donors, and we assessed the proliferation of T lymphocyte cells in the presence and absence of pregnenolone sulfate, one of the detected steroids and the main precursor for all of them, using flow cytometry. What we noticed is a significant drop in proliferation of T lymphocyte cells, and also a significant drop and reduction in cytokine secretion. In summary, in this study, we report longitudinal changes in host steroid biosynthesis and associated transcriptional changes in immune cells in two different ethnic groups. And we identify immunosuppressive role of endogenous steroids in Plasmodium falciparum malaria. Finally, the results of this study have been accepted for publication at Nature Metabolism and will be available at the provided link starting June 11th. Thank you very much.